Very good evening. This is Jeevita from Team Gene Approach. Hope everyone is fresh, had your evening snack, and ready for the webinar. So, firstly, Gene Approach is an edutainment startup where we basically provide education along with interactive activities related to science. So, the ideology behind our team is we are all MTech genetic research scholars. So, we wanted the world to know the importance of science and the basic roots. So, as Ellen Ullman says, genetic is where we come from, and it's deeply natural to know what it's happening. So, following him and sticking to the motto of this, let's move forward. So, today we have our guest speaker, Mr. Sabari Krishnan. He is a genetic engineering gold medalist graduate and a freelance educator. And recently, he attended as a guest speaker to give awareness about the menstruation to the teenage girls in the higher secondary school, Thiruvananthapuram. So, the team Gene Approach is extremely happy to introduce him to you through this webinar. Hope we will be having an enlightening evening ahead with Sabari Krishnan. So, over to you, Sabari. Good evening, Sabari.
Yeah, good evening, good evening.
Yeah,
So, is my audio clear?
yeah, everything is fine.
All right, good.
Yeah.
Yeah, so sorry for, I don't know, some technical issues were there, so I couldn't reach for the introduction. All right, so without further delay, let me start. So, hi, good evening, everyone, and welcome to this wonderful webinar conducted by Gene Poach. So, myself, I'm Sabari Krishnan. I'm a genetic engineering graduate, and now I'm doing a freelance tutoring thing to help students. So, Jeevitan, may I present my screen?
Yes, Sabari, sure.
Okay, thank you. Okay. I hope everybody could see my screen. May anyone please let me know?
Yes, sir, we can see.
Good, awesome. All right, so as you know, the topic for today's webinar is genomic DNA isolation. So, actually, genomic DNA is a magnificent, wonderful subject if we start exploring. But have you ever imagined we can isolate that super molecule which determines the code of life just at home? But even if you are preparing a recipe at home, you should know what all are the basics that or what all are the principles behind that. If you add some masala to a curry, you should know how much is the spice level of the masala, right? So similarly, if you are isolating genomic DNA, you should be knowing what particularly each solution that you are adding does to DNA and how we'll isolate it. So that is what mainly we'll be focusing in this webinar. Now, before going into that, first step, you need to know what is DNA. That is the basic idea about what DNA, what is DNA made of, and so on. And the types of DNA, you need to know which type we are focusing particularly in this webinar, and then we'll be seeing how to or what are the principles behind DNA isolation, what we do in lab versus what we do in home at home. Then some laboratory techniques, I'll go across that. So I came to know from the organizing team that you're all prepared very enthusiastically for this webinar with all the tools required to isolate DNA. Well, it's a very simple thing to do, but knowing the principle behind that is the main aim of this webinar. All right. So let's move ahead. So what is DNA? You all know what is DNA. It is deoxyribonucleic acid, right? So yeah, this alpha helical molecule is DNA. Now, it is composed of two polynucleotide chains. So you can see that, just a minute, let me, yeah. Okay, so you can see there is these two polynucleotide chains are there. So what is a nucleotide actually? So these are all polymers. Polymers have their subsequent units are known as monomers. So similarly, DNA is a polymer, and its monomer is known as a nucleotide. And this is what a nucleotide looks like. These are all basic information, but this will be very helpful to know the principle behind isolation. Why? We'll come across that soon. Now we have sugar, which is, yeah. Yes, Karthikeyan. Sorry? Um, all right. Okay, right now, may anybody tell me what are you seeing right now? Oh, is it so? All right, all right. Maybe some network issues. Just a minute. Okay, now, okay, what is it now? Anybody? What is DNA? Fine. Okay. So are you fine if I present like this? I mean the slides in the side, and if I think if I go to slideshow mode, then I guess the problem is there. I guess, is it fine? Yeah, I was sharing the entire screen in the slideshow mode, but I guess it's because of that you couldn't see the slides moving. Maybe. Okay, I'll just go ahead. If you find any difficulty, please let me know. All right? Okay, thank you. Thank you. All right. So, yeah, I was talking about nucleotide. Nucleotide has a sugar molecule as its core, and it has a nitrogen base. Nitrogen bases are those are organic molecules, which is actually the heart of a DNA. Or actually, the function of DNA is to store genetic information, and all these information is stored in the form of these nucleotides. All right. So we have adenine, thymine, guanine, cytosine, four nucleotides in DNA. Then at the other end of the sugar molecule, we have a phosphate group, uh, which is actually it is known as the sugar phosphate backbone of DNA and the bases. So you can see all these ladder-like things in DNA. So those are the hydrogen bonding between the bases, and you can see the helices or the backbone-like structure, which is made of sugar and phosphate group. So it contains, so DNA is basically what? It transmits genetic information from one organism to another, or from one generation to another, right? And whichever life form you take, it always will have a type of nucleic acid. It can be DNA mostly in all higher organisms and lower organisms, or it can be RNA, which, yeah, we are fearing about coronavirus right now, right? So it has an RNA genome, all right. Anyways, it is these are all nucleic acids, and nucleic acids code for life. Fine. Okay, now we'll be moving to the types of DNA. So uh, we may be wondered that do we have only just one type of DNA, or do we have more than one type of DNA? The question is very legit, because DNA should be only of one kind, but in our body we have more than one type. The first is the nuclear DNA, which we'll be exploring today, and we'll be isolating it right now. Okay, and so we have genomic DNA, that is the nuclear DNA. That means inside a cell we have a nucleus, and inside the nucleus there is a genetic material, which is the main genetic material which makes our body, okay, which codes for protein and makes the body as it is. So it is known as genomic DNA. So it is found enclosed in the nuclear membrane in eukaryotes and in the nucleoid region in prokaryotes, because prokaryotes don't have a proper nucleus, and the what that genetic material is known as nucleoid. So that is with nuclear DNA. Now, coming to the next type of DNA that we possess is extranuclear DNA, and in us, it is found in mitochondria. So there was a very interesting study with mitochondria that mitochondria, I mean, during the beginning of life, only prokaryotes existed, and in some stage of evolution, a particular cell engulfed a prokaryotic bacteria like cell, all right. But somehow this bacteria didn't get digested. It somehow lived inside the consumer cell, fine, and produced energy. And over time, this prokaryotic bacteria over time evolved into mitochondria and chloroplasts. So the plant kingdom has invited this new guest as chloroplast to harness energy, and we heterotrophs, or the animal kingdom people, has invited this special guest as mitochondria to harness the energy. So it is a symbiotic relationship. We give them nutrients, we take back energy. So without these prokaryotes, we wouldn't have evolved into multicellular organisms. So that DNA is also very important because it gives energy to us. Okay, so those type of organelles, those organelles contain their own DNA, which is known as extranuclear DNA or extra chromosomal DNA, which is basically found in mitochondria and chloroplasts in eukaryotes. And in prokaryotes, in bacteria too, there are certain circular DNA which eventually came into mitochondria. These are known as plasmids. Anyway, we are not going to details of that. The last type, which is a very basic type, is known as free DNA. Actually, these are free or fragmented DNA. This can be found in our blood, but at a very, very minimal level, because cells grow, cells die within seconds. So the byproducts of that may come to the blood, escaping our strong filtration systems. So it can be out. Plus, it can be in the environment. Actually, there is a series in Discovery Channel which is related to finding monsters. I mean, the Yeti, then the dragons and all. Okay, so what they do is they go to the places where people claim that they have found these things, and they collect the samples, natural samples such as water, flowing water, or the trees there, to find evidences of some different DNA compared to the other animals in that area, like that. So these are all very interesting areas in biology to explore. Okay. So, anyways, today's topic will be nuclear DNA or genomic DNA. Now, genomic DNA. Going into genomic DNA, may I please narrow down this to human genome? Because, anyways, ultimately, all these genomic studies reach human genome or for the welfare of humans, right? I'm not being so humanistic, but yeah, basically all these studies are for the welfare of humans. So to know that, what we need to know is how our genome is structured inside our cell. So in our cell, we all know that there are DNA strands known as chromatin. So during mitosis stage or during any stage of cell division, these chromatin fibers condense to form chromosomes. Nothing different. These are also DNA coupled with protein molecules. So chromosomes are basically condensed DNA. Now, in a cell, in any human cell, inside the nucleus, these chromosomes are 46 in number, 22 pairs plus one pair. These 22 pairs are known as autosomes or the normal chromosomes, which code for all the genes and everything, whatever proteins you need. plus the last pair, which is known as the allosome or which is known as sex chromosome. Basically, these also code for a variety of proteins, but basic function of these is to determine the gender of the individual. So what you can see at the right is known as a karyotype, which is this is an ideogram, which is a precursor of a karyotype. Karyotype is the original arrangement of chromosomes, but this is an ideogram. This serves as a reference to which all chromosomes should come in place. Well, these are all clinical terms. However, these are the chromosomes found in human cell. So all the 23 chromosome pairs, 22 plus 1. Now, including these 23 pairs of chromosomes and the mitochondrial DNA. So mitochondria actually doesn't have a repair system. So if you track down each mitochondria, the DNA may differ in each mitochondria. So it is a very tedious job to code all the mitochondria, right? And every time cells keep dividing, mitochondria keeps dividing independently. Plus the chromosomes. You don't know what type of DNA mutation has happened in your heart cell or in your liver cell or in your kidney cell, right? So you don't know. But however, this is an ideal thing, ideal term, human genome. It is a collection of all, the entire DNA which is present in all your cells, plus all your mitochondria. That is human genome, all right? That is the definition of human genome. However, we take only some particular cells and then we sequence it and then we claim it as human genome. Fine. For technical easiness. Anyways, human genomes or human genome is a DNA. It is DNA. Now, DNA, the main function is to code for protein, but there are several million base pairs of DNA which doesn't code for any protein. These are known as junk DNA. We just, yeah, termed it, but now the hot spot in DNA research is to decipher what is happening in this junk DNA. Nature doesn't give this overload of information without any purpose, right? So there should be some purpose and scientists are behind that. So there are two types in that way: protein coding DNA genes or DNA and non-coding DNA. Okay, protein coding, it codes for a particular protein which has a particular function in our body. Non-coding, it stays as such. As of now, we only know that, but it has other purposes also. Maybe it will enhance a protein production, something like that. Well, research is going on. Now, haploid human genomes. Now, the term haploid, diploid, it is a very basic genetic term. We are all diploid organisms. That means we are all products of sexual reproduction, right? That means half of our chromosomes comes from our father and half from our mother, right? Now, so one part from father, one part from mother. That's why we have 46 chromosomes or 23 pairs, 23 into 2. Now, this is why we are known as diploid chromosome. Di means two. There are haploid organisms which basically are a result of asexual reproduction. Sorry, asexual reproduction. That means they just divide. They don't want to copy or they don't want to what segregate their chromosomes, then recombine. They just divide. Now, if we consider a haploid cell, maybe the gametes, okay? Haploid cells, the human genome has 3 billion base pairs, that is 3 10 power 9 base pairs. And what makes each one of us different is just 0.1% of that. That means 3 into 10 power 8 base pairs. Those many base, those the changes in those many base pairs is what makes each one of us individually. Imagine how incredible is that, right? So there are various levels of this, because if we go to places like Mongolia, China, Japan, and all, these the facial identities, the changes in the facial identities are, we feel that it is minimal. But if we take India, the facial features vary. So that's why I told these levels are different. Um, the changes may be happening in different regions of the DNA, but ultimately it is estimated as 0.1% with single nucleotide variants. I mean, with one base pair changes, all right? Or zero point 0.6 when considered with indels. Indels are nothing but insertions and deletions. Well, we can cover these topics maybe in a future webinar, because these are very extensive topics, okay, and very interesting also. Anyway, now the question comes, okay, you have been blabbering about DNA and all these, but why? This question will be there in your mind, right? Now, this is the answer. Well, basic thing for genetic studies, okay? You need to learn a cause of a disease. Suppose somebody is bleeding heavily and it doesn't clot. You need to know what happened. Now, you're going to track down and found that it's a genetic cause. What is that disease? It's known as hemophilia. Okay, this bleeding disorder is known as hemophilia if it is genetic. Now, such that, like that, you need to find the genetic cause of some genetic disorders. Then you need to purify DNA, you need to sequence DNA, only then you can confirm it. These are confirmatory tests, all right, sequencing DNA and all. But before anything, if you want to touch DNA, you need it in a pure form. And for that, you need to isolate that. Fine. Then if you want to develop drugs, so now you have Covaxin, Covishield, Pfizer, uh, uh, like it's a what? Pick from the shop scene now, right? Whichever vaccine you want, you can. But everything has its roots in isolating the particular nucleic acid. For coronavirus, it is RNA. So you isolated RNA, then you sequenced it, then you using various computational applications. Well, this is also a topic of immense acceptance from the public. It's a very interesting topic, computational drug approach. All right. So using those, we found the targets or particular proteins, or we developed antibodies against that particular region of the virus, right? So anyways, it started all with the nucleic acid. For virus, it was RNA, fine. So these are the applications which, or these are the reasons, main reasons why we need to isolate genomic DNA. Then if we, for forensic sciences, if you have some samples, blood or hair or something like that from a crime scene, using a very interesting technique known as DNA fingerprinting, we can track down to the criminal or something like that. Okay, we can solve that case. Or for paternity issues, if we don't know who the parents are of a child, or it doesn't apply just for humans, for animals also. We employ this technique, and all for all those things, we need DNA or the subsequent nucleic acid, because it doesn't lie. That's the reason. Anyways, moving on. So this is genomic DNA isolation, the basic steps. I was thinking about another thing. Why don't we complete the isolation, then go with the basic steps? Um, Anybody from the organizing team? Because, hello?
Yes.
Yeah, oh, thank you. Okay, so no, I was thinking, why don't we go with the nucleic acid isolation, then go with the explanation? What do you
Yeah,
say?
okay, Sabari. If everyone is ready with the requirements, we can continue.
Okay,
If
okay,
anyone
fine.
is performing from their side.
All right, all right. Okay, then I'll do a thing. I'll just start with this so that
Okay.
it'll give them ample time to collect the things. Fine.
Okay. Okay.
Okay, fine, fine. All right. Okay, so everybody, please get ready with your equipment, your instruments, so that we can isolate DNA right now. Okay, so while you get into that, I'll just tell you what we are going to do right now in scientific terms a bit. So the first step is known as lysis. Lysis is nothing but crushing everything and making into a, you know, puree form. Okay, considering the fruit that we are taking. Then the second step is known as separation, where we separate all these cell debris, the seeds, the skin of the fruit, everything from the mashed pulp, which is actually DNA plus protein plus lipids plus so many macromolecules. Okay, we'll just filter it. Fine. That is the thing. Third step is known as precipitation, where we remove unwanted things chemically. Okay. In the second step, separation, we removed it physically using a filter, using a cloth. Fine. But in this step, we'll isolate it, we'll separate it chemically. That is using different solvents. Okay. Then we have purification, which is basically the purification of DNA using solvents. So it is actually a play of chemistry here. The solvents and the solubility, that is what determines good quality DNA. I'll go through each step when we do genomic DNA isolation right now. So I hope everybody is ready. Let me just shift my camera to the position. Okay, just a minute. Yeah, should I wait for some time or shall I just start?
You can proceed, Sabari.
All right. So here we have a work table. Yeah, it's at home. Now, I highly recommend using gloves because there are corrosive materials. Those are detergent and salt. Fine. Maybe it will be sensitive for people, but however, first step, as I told, is lysis. Now, lysis is, I don't know you could see clearly, but this is the fruit which I collected. These are known as mulberries. Now, the selection of fruit is important because if you choose hard fruits such as guava or apple with its core and skins, okay, if you choose those, then mashing it will be very difficult. So you want to choose berries. It will be the best, like strawberries, blueberries, and all. Okay. Now we have this. So what I'm gonna do is I'm gonna mash this. So that will be the first step, which is known as lysis. So I'm gonna transfer this and using a spoon, I'm gonna mash it. Fine. You can just use your hand, but if you're using it or if you're trying this to do in your lab, then it will be a big problem because instead of your or with your fruit DNA, your DNA may also be mixed, and but it opens a very huge possibility that you can make what fruit man hybrid, something like that. Okay. Well, these are things which people research on, so there is a very good opportunity and scope for that. Well, fine. So we have our DNA, our fruit roughly matched. I don't know whether you could see this because lighting it's evening here, right? Mhm. Well, it's it mashed. Now the next step is we are gonna add some water. I have some water here. It's better this water is cold, not ice cold, it's cold. Now, the moment you mash this fruit, the cells break, right? That's what lysis is. The cells start to break. When these cells break, it is a natural instinct of any cell to avoid harming other cells. So it starts releasing certain enzymes. Certain enzymes start gets activated and tends to eat whatever DNA is released. So those type of enzymes are known as DNases. Okay, so DNase is one particular thing which we call cofactor for the enzyme to get activated is magnesium ions. But anyways, at home we are not going to try to remove that. Instead, what we'll do, we'll just reduce the temperature. By reducing the temperature, what we'll do is the enzyme activity will be slowed down. So we'll have more time to work with our DNA. Anyways, now we have this mashed thing. We are going to separate it and separation by filtration. Okay. So I have a tissue here. So what I'm going to do is, ideally you need to use a cheesecloth, but fortunately I don't have that. What I'm going to do is, I'm just going to, all right, I want to show this thing. This is known as a falcon tube. It is basically used for n number of experiments in lab. Fine. I got this from my lab. Yeah. Okay. So I'm gonna put this tissue paper on the mouth of it. Okay. I'm just gonna pour some of the isolate through the tissue paper. Yes, the tissue paper should be dampened, but it will be better if you could just squeeze out whatever liquid is coming. It will contain some cells. Okay, fine. So yeah, squeeze and it is clear. Yeah, one seal is there, but it's clear. So it's almost 2 ml of the isolate. Fine. Well, for the time's sake, I'm just speeding things up. Right, but please take your time to understand what is going on and I'll be explaining it soon. So it is nearly 2 ml of the isolate. Fine. Okay. Now, next step is separation. So going on to separation, let me just move to the principal screen. Okay, moving on to the separation. It is isolating or separating the nucleic acid from the proteins attached with it. Now that is done by adding a detergent. Now detergents have a unique property. It has a hydrophilic end and a hydrophobic end, right? So the hydrophilic end always binds with water and its ions. Okay, but the hydrophobic end always binds with the protein because certain amino acids of the protein, not all amino acids, but certain amino acids of the protein, which has, see, now the color change. Well, this is also an indicator. Mulberry is a very good indicator. Okay, now the color changed to black. Well, chemistry. Fine, anyways. Now I have added the detergent. So what the detergent does is it denatures the proteins which is attached to the DNA. Okay, so that is what the detergent does. And it breaks the cell membrane because cell membrane is also basically made of detergents, right? Because those are phospholipid bilayers, lipids. Detergent is also a type of lipid. Fine. So lipids and lipids come close, they solubilize, they dissolve the membranes. So that is what it happens here, and it is happening here. Okay, the color change is actually because the certain compounds in mulberry is acts as an indicator to high pH because these detergents are high pH. Okay. Okay. Anyways, we have that. Now remember, don't shake too much with your detergent. It start to bubble and it will be very difficult to see the DNA precipitation. Now that we have added this, next chemical that will be added is salt. Not just normal salt. I mean, it is table salt, but saturated sodium chloride solution, which is also known as brine. Now, what this sodium chloride does is it is also in okay, two more. So okay, I didn't tell you the measurements, right? It is ideal to take 2 ml, I mean the same volumes of the pulp or filtrate, detergent and salt to get good results. But again, it depends on the concentration of what we have made, the chemicals. Now, yeah, so we have added, let it sit for 1 or 2 minutes, and I'll tell you the story of why we have added all these bunch of chemicals. Fine. Okay. What is happening is lysis has happened. We have physically disrupted the cells and the tissues. Now, with that physical destruction, we got a liquid. Okay, we transferred it into the falcon tube. Then what we are doing is we added detergent. Now detergent in lab we use detergents such as SDS. SDS is sodium potassium sulfate, and CTAB. It is another, and it is well famous because it's CTAB, which is cetyl trimethyl ammonium bromide. Don't want to learn these terms. These are basically detergents with various properties. Fine. Now, after this lysis, what we are going to do, so during the lysis the cell membranes has broken, the tissue matrices, certain other proteins have dissolved, everything has dissolved and it's made into this slurry. Fine. Now, by adding certain reagents, we'll separate the proteins from the DNA. Now, this is completely a laboratory procedure. One, because we don't have access to those chemicals at home. Second, this is just a crude isolation of DNA. Anyway, we cannot use this for lab purposes. Fine. It's not at all pure. Fine. It may be like 40 or 30% pure. We need 99.9% pure DNA for sequencing studies. So imagine how much work it should be done there. Okay. Anyways, so lysis is there. Separation, it is done with an organic solvent such as 2524-1 PCI, which is phenol chloroform isoamyl alcohol mixture. So this is a organic solvent. Now, talking about the chemistry behind that, uh, proteins have certain hydrophobic and hydrophilic amino acids, and they tend to solubilize with their like amino acids. It is a rule in chemistry. Like, three likes, right? Yeah, it is a rule. By that I mean, um, oil always tends to mix with oils, right? Oil doesn't tend to mix with water because their natures are different. Oil is hydrophobic, and water is, yeah, water itself is, yeah, water, hydrophilic, right? So it doesn't tend to mix. So this very basic nature is exploited here. So by adding this organic mixture, all these proteins are drawn into the solvent that we add. Then we take a centrifugation step where we separate all these, but we cannot use it right now. Next step, yes, it is precipitation, and it is the most interesting part because right now in this falcon tube, all the cells have been broken down, all the proteins have been coupled with the detergents, the lipids have been uh, trapped by the detergent. Then the salt. Now, salt, I'll just come to it soon. Why we add salt solution, the brine to it. But before that, you need to see the DNA coming out. Okay, this, what I have here is ice cold isopropanol. It is best to use isopropyl alcohol, isopropanol. Well, you can use alcohol, ethanol, basic ethanol, your sanitizers, okay, those are 70% ethanols, but the results may vary. The best is isopropyl alcohol. You can also use nail polish remover, which is basically acetone. So any organic solvent is fine. Now I'll tell you the chemistry, why we are adding an organic solvent. Now, how we'll add this? This is a very important step. You need to add this alcohol through the sides of the falcon tube, not just splash around because, yeah, you can't see the DNA coming out if you splash around. Fine. You need to just trickle the alcohol through the sides of the falcon. Just maybe like 2 ml or so. Fine. You need to have a transparent layer of alcohol and you can see small threads of DNA just precipitating, just coming out of that dark layer into the isopropanol layer. Slowly, gently, you need to trickle through the walls of the test tube. Okay, so here we have our DNA. Let it sit for a while, okay, so that most DNA will get precipitated. Let it sit without any disturbance. Back to science. Yes, 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 back to science. Now, why we have added all these? It's a mess here. Anyways, why we have added all these? Yes. So by adding sodium chloride, what we have done is, do you remember that nucleotide is made of, it had a phosphate group? Do you remember that? Okay. This phosphate group is what makes DNA soluble in water. DNA is soluble in water. All right. Now this is what this phosphate group interacts with the water ions, hence showing its solubility in water. But by adding sodium chloride, what we do is these sodium ions in the sodium chloride solution, they go and attach themselves to the phosphate ions because phosphate has three negative charges in them and sodium has a positive charge. So sodium ions go and neutralize these charges by binding with them. Now it is everywhere, right? If somebody is not working fantastically, they'll just throw you out of the whatever organization, something like that. Fine. The same happens here. Now, water is this DNA is not interacting with the water because their charges are neutralized. Now, what happens? Water cannot hold this DNA and it just drops. Now, to enhance that dropping out of the solution, we add isopropyl alcohol. It has much more receptivity of molecules, especially these precipitating molecules compared to if we add water, it doesn't, it just mixes up. Okay, but if we add alcohol to this solution, the DNA just comes out of it. Now, why? Why we are adding ice cold? Precipitation and reactions, chemical reactions, are completely opposite things to some extent. Precipitation is leaving a molecule or extracting a molecule out of a solution, while reaction or preparing a solution is it is just a play of temperatures because as the temperatures rise, solubility increases, and as the temperature decreases, precipitation increases. Okay, so first we have already debar the DNA out of hydrophilus or the hydrous solution because these are all water loving in nature, right? So all this DNA was interacting with nature, but by adding sodium chloride, adding the sodium ions, the DNA has been neutralized and now it doesn't have any place in that solution. So it is trying to come out. By adding the alcohol, we are enhancing that DNA to come out. Okay. Now, why chilled alcohol? Because again, we are enhancing the DNA to come out because of low temperature. Okay. This is the basic idea of the steps that we have done now to isolate the DNA. And yes, I don't know, and I hope everybody could see small white strands of DNA. I don't know, you could see clearly. Small white strands of DNA. Well, I'm not claiming that as DNA alone, but small white strands you could see, right? You need to experiment. If you don't get once, you need to do until you get thrice. That is a basic rule. Yeah. In labs, you need to do like that. So you could see the strands of DNA and some bubbles coming out of the dark colored solution, which is up till here, and the alcohol solution, alcohol layer. So you can see the white strands here. Now, if you could see very good white strands, lot of white strands, then using a toothpick, you could just swap. Yes? Sure, sure, sure. Um, I'm not sure. Um, okay, these are the strands, but well, I'm putting, I think it is connected, but I'm afraid that you cannot see clearly. You can see white things here. Yeah, I know you couldn't see, but all right. Well, it is a thing that you will see, but now due to some technical difficulties, I need to adjust the cameras and lot of things. Oh, okay. All right, I'm back. Fine. So this is the thing. We have isolated DNA apparently, and I request everybody, those who got, congratulations, because you are at the starting point of any experiment with DNA. You should understand the principles rather than just mugging up the entire procedure. You need to understand the principle. Only then you will be knowing why we are adding all these chemicals, how the DNA comes out, what is the nature of DNA. My professor in my lab always told, so he was a cytogeneticist. He used to do cell culture things. So what we do is we do a cell culture, then we just kill it. Yeah, that is the thing. We kill it. But what he says is, even if we are handling with non-living things, just try to feel them. Then your experiment will be 100% correct. So that is what I'm saying. Just go with the principle thoroughly rather than mugging all the measurements, the volumes of the liquids that you want to know. And that's what basically this webinar is about. So the four steps: lysis, separation, precipitation, purification. Yes, purification we are not going to discuss now because it requires much more. Yes, Janardhanan sir, I'm coming to the purification step. Purification is actually a step which requires high precision and some enzymes. Because what you isolated right now is not just genomic DNA. We can say it is nucleic acid. Because we know in a cell there is DNA, RNA, then since it is a plant, it's a fruit, but yeah, it's a plant. So it may have chloroplast DNA also, right? So it is a mixture of various nucleic acids. But if you want to sequence DNA, you just want DNA, just genomic DNA. not even mitochondrial DNA, not even any RNAs, right? So what you'll do to remove this RNA? We can't fish that out because all are nucleic acids. So what we do is, after multiple steps, so once this DNA is isolated, we'll centrifuge it to pellet it down so that we'll get a solid state of this DNA. Then we'll wash with ethanol, 70% ethanol. Then we won't touch water. Okay, we won't touch this with water because DNA will tend to solubilize again. And when, just remember this, whenever a solid or a soluble thing such as salt starts interacting with water, its half-life will come down. So DNA apparently it has a half-life of 512 years. It is a very long half-life for a molecule. But once you start mixing DNA with water after pelleting it, after resolubly, soluble, I mean resolubilizing it with water, then the quality of DNA which you are looking, it will come down drastically. Then you cannot use that for any sequencing techniques. All right, so keeping that in mind, purification is a very important procedure. We'll only use ethanol. Now, to remove this RNA, the disturbing molecule, yes, RNase, an enzyme which is ribonuclease. Okay, we'll add that so that that enzyme will just eat up whatever remaining RNA is there. It will just degrade. But again, there is a problem. By adding RNAs, you are introducing an enzyme which we don't want, right? Now, all these things should be there in the mind while we are working with that, working with DNA. Now, two more slides to go. I guess we have time. That is measuring your DNA. Now, you have isolated your DNA, but do you know how good your DNA is or how much you have isolated? Now, these are entirely laboratory principles and procedures, but you need to know that only then we can isolate good quality DNA right from the start. Fine. So the first technique is qualitative, or the first set of guidelines which you need to follow is the qualitative thing and the quantitative analysis. First, we go for qualitative analysis, where we check the quality of the DNA, whether the DNA has some contamination in it, or whether the DNA is degraded to some extent. We see those things. And that is done by a technique known as agarose gel electrophoresis. And the second technique is known as quantitative analysis, where we measure the amount of DNA and using a spectrophotometer. So this is the setup of agarose gel electrophoresis. So what you do is you have your isolated DNA. So this is what we get in lab. It is entirely different from what we got in home, right? But again, the principles, procedures, everything is similar. Just the reagents, the quality of reagents and the working principle, working instruments make all these difference. So you can, I don't know whether you could see it, but if you quickly go to the first slide, you can see these white strands. This is what exactly genomic DNA is, right? So this quality genomic DNA we are looking for, what we do is we mix it with a dye such as bromophenol blue. It is a blue colored dye, all right, and a fluorescent dye which is known as ethidium bromide. Unfortunately, this is a carcinogen. It causes cancer. But why we add is we cannot see DNA, but ethidium bromide has its property to bind to DNA, bind to the strands of DNA. This bromophenol blue just gives an idea where the DNA has reached in this particular procedure. So I'll tell you the procedure. So this is basically a electrochemistry setup. So what we do is DNA, we know it is negatively charged. After all these washing and all the sodium ions, everything has been removed. Fine. Now DNA has regained its negative charge. Now with this negative charge, we are mixing with this DNA. We are mixing chromophenol blue, some weighing agents such as glycerol to weigh it into these slits. These slits are known as wells technically, and this blue color film that you see is actually a gel which is made of agarose. Agarose is a polysaccharide. Now the structure of agarose is very interesting. It is like a mesh. So when you load your DNA into these wells and make sure it is in the negative side, because our aim is to migrate this DNA across the gel. Now, if you need to migrate this DNA across the gel, across an electric field, then negative particles should be there in the negative side, right? Only then it will migrate to the positive side. So, the wells should be fixed in the negative side, negative electrode, and the genes and the DNA will be there in the wells there, happily sitting. And once we add electric component to it, once we switch on the power supply, this DNA will be migrating from the negative electrode to the positive electrode. We'll run it for maybe 50 minutes or up to 1 hour, depending on the concentration of the agarose gel. There are various other techniques to that, but I'm limiting until here. So yeah, this is actually what you see under UV light. So I told ethidium bromide will be added, right? So ethidium bromide, it fluoresces under UV light. So once your DNA has completed its migration across the gel, and you will take the gel off and you will put it under UV light, you will see orange colored bands like this, and all these represents DNA. Well, unfortunately, this DNA is not that good. How do you identify that? Because you can see a smear, right? So smears, if you find in agarose gel, then it's a red card for you in labs. Yes, smears are degraded DNA. You want to see DNA like this, like this. By that I mean these individual bands. So this is what we call a ladder. A ladder is just a reference to identify what type, what is the size of the DNA. Well, these are for another talk maybe. And once you view under UV, you will take a picture, take a photograph. So we have an instrument for that with a camera attached. So you will take the photograph, then you will work with the data. Fine. So this is qualitative analysis. Now, quantitative analysis is it's a bit tricky, but I'm not going into the techniques of technicals and calculations of that. Just explain. So UV visible spectrophotometry is the technique that we use in purity analysis or the quantification of DNA. So what happens, or the basic principle is we illuminate a particular dilution that we have taken from the DNA that we have taken, that we have isolated. We'll illuminate this DNA using UV light and visible light. UV visible, it's a mix. Okay. Now, for DNA particularly, 280 nanometers, 280 nanometers of wavelength is illuminated, particularly to this sample. That will be done by the machine. You can see this process. So from the light source, a lens focuses on to a diffracting prism. Then from the prism, wavelengths are generated, and from the wavelength selector, the particular wavelength is illuminated into our sample, right? So this wavelength selector will set it into 280, 280 and 260. two wavelengths, we'll take a ratio. Well, I'm not going to the techniques of that. So you have the solution here. So you will illuminate it with a particular wavelength. Then that light will obviously pass through the solution, right? But some amount of that light will be absorbed in the solution. So how do we measure that? We'll measure the outcome radiation and we'll compare it with the incident radiation. So this ratio is what determines the purity of DNA. So for DNA, the purity should be 1.8. If we take this ratio, it is the absorbance of 260 nanometers to 280 nanometer absorption. If we take a ratio of that, then for a pure DNA, it should be more than 1.8. I mean, 1.82 is the purest DNA. Okay, so this is with quantitative analysis. So what you have done, you have isolated DNA, you have analyzed its quality, then you have analyzed its quantity. So these three steps is the basic step wherever you go across the world. If you are working with DNA and if you are interested in working with DNA, you should know the basic principles behind genomic DNA isolation, then agro-cell electrophoresis, and UV visible spectrophotometry. Well, unfortunately, we couldn't cover the principles of agro-cell electrophoresis and UV spec, but I hope genomic DNA isolation was clear for you. And that's all.
So, hello everyone, this is Devadarshini from Gene Epoch team. So, we would like to thank Sabari Krishnan, our today's speaker, for being committed and engaging us the whole time with a knowledgeable webinar presentation. We hope you people had a good time and enjoyed the session too. Thanks for being cheerful and an entertaining audience. As the saying goes, successful persons are not gifted, they just work hard, then succeed with success. So, I would like to extend my special mention to the whole team who worked behind the screen, our board members of Gene Epo, Hemanathan, Muhammad Shaibi, Rahul, Sarvesh, Harris, Akila, Sarat Chandrika, Harini, and Satamai. Thanks for your phenomenal work and effort. This is for our growing audience. We hope for you to show your presence in the upcoming events of ours too. We will be releasing our upcoming events very soon. Until keep supporting and stay engaged with us via Instagram, Facebook, and WhatsApp and social media platforms. And we wholeheartedly pleased to have such a great audience as our participants. Thank you for your valuable contribution and for making this webinar more successful. Once again, I thank you all for being here. Thank you.